Welcome to Myeloma 2016. So this year we have the meeting at Boston, and we just completed a very exciting sessions discussing genomics and the current status in 2016 in terms of what we have learned from it for myeloma. And joined by me is two of the panelists from today's session, Gareth Morgan from University of Arkansas and Michele Cavo from Italy. So maybe I'll just start by getting the opinion of Gareth. You know, the data that we know from today and previously in terms of genetics of myeloma is that it has a very important branching kind of evolution, therapeutic selection. But where do you see this going in terms of how we're going to be eventually using this in the clinic for the treatment of myeloma patients?
So it's an apparently simple concept that really we've known about for years, but becomes more and more important as you start to think about how you use therapy. So it tells you that if you want to cure myeloma, you should use combinations of drugs to overcome the interclonal heterogeneity. Mm-hmm. It also tells you that if you do a mutation test on one site, it might be different at another site. So if you use targeted treatment, you can eradicate the disease at one site, but you may not see a response at the other. So it's very important as we move to using targeted treatments and designing combinations of targeted treatment that I think this evolutionary theory comes into its own.
But do you think at present time we have a good enough idea on what are the kind of treatment we need to give to target the different kind of clones? So
we're trying to develop these ideas. So it's clear that RAS and BRAF are key drivers of the disease. If you target BRAF, the tumor cells die. What we understand though now is that there's differential responses. So rather than simply using BRAF, you're going to have to use combinations and understand why some sites respond and others don't. And it could be the presence of that lesion, or it could be another lesion that needs to be targeted. And so I think we're in a very exciting phase of using umbrella study designs, designing biomarkers to enter people into the studies, and then investigating in depth what happens and using that information to better design how we treat people.
Great. Maybe I'll turn to Michele, whose talk is on P53, which is clearly a very important group of myeloma with very high-risk disease. So again, you showed that our current understanding of the role of P53, is still evolving because, the spectrum of abnormality is now slowly, being elucidated. But yet, some of the therapy that seems to be working for this group of patients in the clinic, such as double autologous transplant, you've presented very nice data that the survival is much better, seems somewhat counterintuitive about why they should work in a population of patients which have abnormalities in the P53 pathway. You would imagine that using alkylating agent at high dose may be not so good for these patients. So do you think that there is a kind of a gap between what we know about biology and how the therapeutic trials are moving, at the same time, in the sense that there's the two are not quite linked, and we perhaps need to understand biology a bit better?
Yes, I agree. Probably there is a gap, and the gap probably is justified by the many mechanisms that may concur to induce loss of P53 activity. Mutations and deletions are the simple one, but many other mechanisms at the DNA, RNA, or even at the protein expression level may concur. The data so far available are related to retrospective analysis and do support the role of proteasome inhibitors, and this may be quite easily accepted based on the knowledge of how proteasome inhibitors works and how are the mechanisms that concur to inactivate P53 by the means of MDM2. I think that probably this is a simple suggestion, and what we need is to design prospective clinical trials for this high-risk patient population, and what we really need to start is to design trials based on different approach according to the different risk of the patients.
Yeah,
So I kind of think there's a really important message that came out of this morning. So it's pretty much known, and Michele did a great job of describing this, that loss of 17p is not a good marker for high risk. So not all patients with 17p loss are high risk. You have to have 2 hits on both alleles, and so it's biallelic inactivation that's important, and probably loss of one allele, a mutation in the other, or loss of both by copy number change is really important. So the message is, when you design a study, you need a good biomarker that really reflects high risk, because a lot of 17p minus patients will be low risk.
And I agree with Morgan, and it is likely that the conflicting results obtained, for example, using bortezomib may be related to the different molecular phenotype of these patients, because as I mentioned, 17p, for example, is frequently monoallelic, but wild type gene may be also affected by mutations and other mechanisms, and it is the reason why the novel agent compounds develop at a molecular level, or even gene therapies have not yet ready for prime time, because the landscape is very heterogeneous.
Maybe I'll ask a question, since we're discussing high-risk patients and how best to define high-risk disease. I would say that actually currently all the tools that we have, they're getting better and better, but they often do not tell us much about the biology, of the high-risk disease, right? We have very good gene expression profile marker, but they're often, not exactly telling us why these patients necessarily have high-risk disease. Some of them may reflect some phenotype like proliferation, but again, that is a very blunt downstream kind of phenotype. So how do we think we can use genomics to better define high-risk patients, not just to identify them, but also perhaps to inform us of some molecular targets that we can use?
So I would argue there are 30% of presenting patients at a high risk. The GP70 will identify half of those, so it only identifies 15% of the total. The other are kind of apparently low risk, but relapse within the first year and a half. Right. And so they're 2 very different groups. There's probably very different mechanisms underlying those things. So I think what we need to do is apply a concerted genomic approach to looking at them. So define the mutations, look at the gene expression patterns, look at the methylation status, and define programs that are deregulated rather than focusing on single genes. And I think that's well within our reach, and will probably come to fruition over the next couple of years.
Yes, it is. I'm sure that the future will be based on NGS and other approaches. So my recommendation to use FISH is simply based on what should be done in the daily clinical practice. But I think that in very few years, FISH probably will be abandoned, and more sophisticated techniques which will explore all the different abnormalities will be in use.
I think one of the key points I want to take on, which Gareth said, I think it's very, very important, is that it has to be a comprehensive approach, because sometimes there is a vogue, say, if next generation sequencing is coming into vogue, people are just going in one direction, but you're only getting one layer of the information, which will not give you the whole picture. So I think having a comprehensive approach to analyzing these high-risk disease, and also reclassifying them more using a kind of molecular-based or pathway-based approach would be more helpful therapeutically, rather than using an unsupervised approach, which always turn up those important transcription factors, I guess.
No, absolutely.
Yeah.
And Gareth mentioned about the 30% of high-risk patients, and it is likely that if you consider also abnormalities of chromosome 1, that is 1q and 1p, you can further increase this rate up to at least 50% of those with the newly diagnosed. Probably you would like to discuss about the controversial role of several of these abnormalities.
No, it's fascinating, Michele. I mean, it's really fascinating, but they really interact together. The 1q, 1p, 17p drive huge amounts of high-risk disease, but not as single agents. As single events, they really don't count for all of that much, and you can overcome them with treatment, especially the double autos. But when they have GP70 high, plus you have those abnormalities, you can almost see different subgroups with different responses to treatment.
The number of co-segregation alterations is strictly related to even poor prognosis, according to the increasing number of co-segregation, and many of these co-segregate very frequently.
So I mean, the bit that I enjoyed about your talk was the idea that you can overcome or specifically target some of these abnormalities. So using P53 signals via a kind of DNA damage, cell cycle arrest, apoptosis response. Proteasome inhibitors signal death via a different way. It could be through endoplasmic reticulum stress. And so if you layer on top of that some of the excitement about CAR-Ts, immunotherapy, and antibodies, I think we could be in a time when you take a P53 patient, enter them into a phase 1-2 study, look at the role of combination proteasome inhibitor, IMiD, and daratumumab, say, and then you may see a very high response, and then it would be good to do a randomized study.
It is likely that our continuous improvement of the genomic landscape and genomic alterations underlying myeloma and also the progression of the disease into different phases according to the recent development of new classes of novel drugs will be combined together and will allow us to specifically target and specifically treat certain patient populations.
So maybe just in closing, if we can get some of your thoughts, each one of you to give us your thoughts on moving forward in the next 5 years. How do you see genomics will evolve for myeloma in terms of its utility in the clinic? Are we going to be completely moving away from FISH and so on to a sequence-based kind of...
So if you look historically when Vince Rajkumar wrote that sort of VAD is dead. Well, I think we're at a time when you can write FISH is dead. You can get all of that information based on a next-gen sequencing approach. You can get copy number, translocations, mutated genes, RNA expression. So really the future's about a myeloma-specific panel that's applied in the clinic and uniformly that we can all agree to. And then if we do that, everything else falls out from that. Targeted treatment trials, improved patient outcome. And so it's a key to the future, and we're just there, I think.
When I left the room for this interview, Leif Bershagel was showing a slide with the future, and FISH was crossed off. So I think that really it will be progressively abandoned in the next few years.
Great. So thank you both for your excellent comments again, and I think it's been a great session. The rest of the meeting is still going on, and I'm sure there will be many good discussions later on today as well.